Everyone. Thanks for getting in on the snow day. So a couple quick things. Remember, Monday is the first exam. I will be in at 7:30 a.m. We are going to have it here in our normal classroom. But I will be in early if you guys want some extra time for the exam. And I do recommend it for this particular one. Again, I've said this a couple times, but this is because of the Punnett square genetic problems, because of the Hardy-Weinberg problems that are on this particular exam, and there's several of each. This is one exam that sometimes takes a little bit longer than some of the others. And I prefer you guys are coming in early rather than missing other classes then. There is a pre-lab quiz, and I think I need to get this posted yet, but there is a pre-lab quiz that is due Tuesday then before you come to lab. There's also, remember, we're going to have lab quiz number 1 also Tuesday in lab. And think of that as a mini exam. You do need to study and prepare for that one. That is then going to be a 15-point quiz. So just some things to keep in mind there. Today, we're going to finish up talking about population genetics. And once we get to the end of that lecture set, that is the end of the exam content as well. And then I'll see if you guys have any things you want me to cover, questions. But I might also go ahead and just start with exam 2 content, which is the next lecture set on species and speciation. For today, we left off last lecture really talking about what Hardy-Weinberg equilibrium is, and then also started doing some math problems that with Hardy-Weinberg and population genetics. That Hardy-Weinberg theorem, Hardy-Weinberg equilibrium, again, what it does is given certain numbers for allele frequencies, Hardy-Weinberg equilibrium is an equation that then predicts the genotype frequencies that would be in the population in the absence of any type of evolution, in the absence of any kind of change over time. So what Hardy-Weinberg is describing in essence is what we would expect a population to look like if it was not evolving for a particular gene locus. In real populations, allele and genotype frequencies do change over time. Sometimes they change very, 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 very slowly. Sometimes they can actually change relatively quickly, though. It depends on the selective pressures. It depends upon the context of the environment and the selective pressures. For Hardy-Weinberg to be absolutely true, for Hardy-Weinberg to be true, and there's no evolution occurring, there's 5 conditions that a population have to be met for absolutely no allele or genotype frequencies changes over time or no evolution. The 5 conditions to meet Hardy-Weinberg equilibrium are no mutations taking place, that there is random mating, not non-random mating. So there has to be no mutations, random mating. There is no natural selection taking place, no gene flow in and out of the population, and then extremely large population size. And we're just going to end up this population genetics unit going through these kind of in more detail one at a time. The first condition to meet Hardy-Weinberg equilibrium, again, is no mutations. If you have mutations, this could be point mutations, gene deletions, gene duplications. Any type of mutations alter the gene pool. What you do with mutations is you create or delete alleles. And then that alters the allele frequencies, which then alters the genotype frequency. So anytime you have a mutation, that is a change in the population, or essence, evolution. Now remember, those mutations, though, when it comes to mutations, sometimes they can then be neutral mutations. Sometimes, and those aren't going to really impact the population, but sometimes then they can be either loss of function mutations, and some mutations are gain of function mutations. Remember, mutations are a random process, and it's going to have a random consequence. Next condition to meet Hardy-Weinberg equilibrium is that there is random mating. That means that every individual contributes equally to the next generation. So when I say this random mating is that, again, every individual contributes equally to the next generation if there is random mating. As long as there is random mating, you don't get changes over time. If every individual is contributing equally to the next generation is also going to look the same in a sense. And so with non-random mating or things like preferential mate selection, this will decrease the random mixing of gametes. So with non-random mating, what will happen is one individual or a few individuals will contribute more to the next generation, but that's a change. Because if they have more offspring, their traits get passed on, their variations get passed on at a higher frequency. Now, with non-random mating, this can be used due to things like sexual selection. Again, this is going to be things that can cause non-random mating. And some of the more common causes of non-random mating. But for instance, inbreeding, this is reproduction between close relatives. This tends to happen, especially in smaller populations out in the environment, especially smaller, more isolated populations. But that can actually cause then non-random mating. Sexual selection is probably the big one, though. Sexual selection is, again, we talked about this a few lectures ago, but this is where typically certain mates display certain traits, and then they actually will then mate more successfully. With sexual selection, there's a few different varieties of this, but I gave you the example of peacocks and peahens. But the peacock males that then have the brighter, larger plumage feather displays, they are more successful in tracking mates, and then they mate more successfully. They have a higher frequency of reproduction. But we can have a number of different things like behavioral displays, color displays. We talked about the male guppies and spot coloration. That is a type of sexual selection where the male guppies that are brighter with more spots are more attractive to mates and mate more often. We talked about frog calls. The louder frogs attract mates more readily and then mate more often. That's all types of sexual selection. Sexual selection, by the way, is very common in any species that exhibits sexual dimorphism. What do you guys think sexual dimorphism means? What's di mean? Two, and what's morph mean? Morphism or morphology? The terms come up a few times. Sexual dimorphism is when there is differences in the morphology or the features and shapes of two different sexes or genders. So species are sexually dimorphic when the male and female genders have clear trait differences. And again, we can see sexual dimorphism. This is a peahen, typically just mostly gray feathers, versus a peacock is the male bird, and that is going to have a much brighter color, much brighter coloration, and then also those large feather displays. That's a sexual dimorphic feature. There's a lot of examples of sexual dimorphism, though. Another condition to meet Hardy-Weinberg equilibrium is that there is no natural selection. Again, any differences in the survival and reproductive success are going to then affect allele and genotype frequencies, and those are going to change over time. With natural selection results in certain alleles, if they have a selective fitness advantage, being passed on to the next generation in greater frequency, in greater proportions. That. Okay, the process of natural selection is going to increase those alleles that confer a fitness advantage, or and then it will cause adaptive evolution. Questions about that? Again, what basically natural selection boils down to is unequal reproductive success. Oops. Okay. But remember, when I say unequal reproductive success, what do I mean by that? I mean that those individuals that have traits that make them successful, they're more likely to reproduce. If they're more likely to reproduce, they have more offspring. Those offspring inherit those traits. Those traits make them more successful and also more likely to reproduce. And as you repeat that over generations, those successful traits will increase in frequency over time. And by successful, again, I mean that they confer a fitness advantage. And what does fitness biologically describe? When I say that term fitness advantage, what do I really mean by fitness advantage? Max? Yeah, fitness is a measure of reproductive success. And really, it just boils down to more offspring, more fitness. Okay, it's a little bit more complicated than that. We're just going to boil down to typically the more offspring that a organism has, the higher its fitness is. That it's all about reproductive success, not necessarily survival. All right. Another condition that is necessary to meet Hardy-Weinberg equilibrium is there cannot be gene flow. So no gene flow. Gene flow is movement of individuals or gametes into or out of a population. So gene flow, we're going to talk about this a few times over the course of the semester. Gene flow is where individuals either exit, or sometimes their gametes exit out of a population, or it can also be when individuals or their gametes enter into a population that wasn't there previously. By moving alleles in or out of a population, you then alter the allele and genotype frequencies. Immigration. This is where genes or alleles move into a population. New individuals carrying genes and alleles immigrate into or move into a population. Emigration, this is where genes or alleles move out of a population. You have a population of individuals, some of them leave and exit the population. That's going to be emigration. Again, it looks a little bit like this. With immigration, you might have, say, this is my island in the ocean. With immigration, you might have a few new individuals that come in and move into that population. Here before, say we had green, red, yellow, blue traits, there was 2 of each. So they were equal. After those new individuals moved in, now we have 3 green, 3 blue, 3 yellow, but only 2 red. So the red trait, in essence, decreased in frequency relative to the other traits, and that's a change in the population structure. E-migration is again where these individuals exit out. Again, we might have an island in the ocean. Let's just say though, there's 4 red, 4 green, 4 yellow, 4 blue. But now some individuals exit and leave that population. But when they leave the population, the genes, the alleles that they are carrying exit with them. Say then a green, yellow, and blue trait individual leave. Now we're left with a population that is different in the structure. It turns out the red trait is actually more common in this case because now it's 4 red to 3 green to 3 yellow to 3 blue. But that changes this. Gene flow is more likely than mutation to alter allele frequencies. Gene flow causes quick or fast changes in population genetics. So these immigration events, these e-migration events can actually change the genetic composition of a population pretty fast. And can cause pretty substantial changes in that population then. Actually faster than mutation in many cases. Also, the more gene flow you have between populations. So if you have a population here, you have a population here, and they're normally not interacting. But if you have regular gene flow between them, it tends to keep the genetics of those two populations similar. So as long as there's gene flow tends to reduce differences in the genetic structure of one population relative to another, as long as they're regularly interacting. That is something we're going to talk more about here in a little bit, though, with speciation. All right, last condition to meet Hardy-Weinberg equilibrium is that there needs to be large population size. The smaller a population, the more likely that allele frequencies will fluctuate randomly from one generation to the next, to the next, to the next. And again, random. Allele fluctuation, that is genetic drift. Again, the smaller population, the more likely that genetic drift is going to cause these random allele fluctuations and these random changes in the population. I used the example earlier when we talked about genetic drift of flipping a coin 10 times versus 1,000 times. If you flip a coin 10 times, you expect 5 heads, 5 tails. But if you then instead get one difference, if you do 4 heads, 6 tails, that's actually like a 20% difference there. So that's a pretty big difference. If you flip the coin 1,000 times, you're expecting 500 to 500. If you get off by 1, that's just 499 to 501, or in essence, a 2% change. So by having a larger population, and the larger it is, the smaller the percent of change that those random fluctuations cause. Okay. Genetic drift is, again, random allele frequency fluctuation over generational time. But again, with that one, the smaller a population, the greater or the percentage of that chain that is made up. Okay, so the smaller population, the greater the chance of a deviation from kind of expected results. And it really just boils down to statistics on that math. Especially in small populations, genetic drift often has the greatest impact by causing allele loss then as well, where you get these random fluctuations, and what ends up happening by these random fluctuations, rare alleles get lost completely from the population, and they in essence go extinct from the population, even if that rare allele was beneficial sometimes. And so that is one of the main ways that genetic drift is going to affect populations, is through just these random changes, but also sometimes through loss of alleles, especially rare alleles. Do you guys have questions about genetic drift? Or do you have any questions then about those 5 conditions to meet Hardy-Weinberg equilibrium?
So it's random mating, it's not no random mating.
To meet Hardy-Weinberg equilibrium, where no evolution is going to occur, you have to have random matings. And one of the things I find if I ask about this, say on the exam, which I probably will, I do find that sometimes one of the more common mistakes is people forget which one you require and which one you don't want to have. So it's no mutations, no natural selection, no gene flow. It's yes to large population size and yes to random mating.
What do you mean by random mating?
Random mating is where every individual in the population has an equal chance of mating. They are all going to have an equal chance of mating. They're all going to contribute equally to the next generation. No sexual selection, yeah. All right, last thing I want to point out with then Hardy-Weinberg, if you depart from these conditions, that will result in evolutionary change. Sometimes slow evolutionary change, but occasionally even fast evolutionary change. And we'll start talking about examples of those. But I also want to point out, natural populations can simultaneously be undergoing evolution at some gene loci, while being in Hardy-Weinberg equilibrium and not evolving then at other gene loci. So for humans, it's actually estimated we have about 20,000 to 25,000 genes. In human populations, we might be seeing evolution at certain ones of those gene loci, but not at others. And that could be happening simultaneously, where an organism, a species, can be evolving at these gene loci, but maybe not so much at these others. Just to kind of give you guys an example, there is one gene locus in humans that is fixed. Do you guys remember what fixed gene locus describes? If we say a gene locus in a population is fixed, what does that mean?
It can't evolve.
Yes, technically, yes. But it's a little bit more specific than that.
There's only one.
Yeah, there's only one allele present for every member of the population. So a fixed gene locus, this means there's only one allele possible in the population. Everybody has homozygous for that exact same allele. In humans, there is a gene that makes a protein called beta-2 microglobulin. Everybody in this class has the exact same version of beta-2 microglobulin. That is a fixed gene locus. But if there is no variation at the gene locus, as Ronnie said, you cannot evolve at that gene locus. If there's only one possible allele, that's it. And there's no evolution present at that particular gene locus. Okay, so beta-2 microglobulin is not evolving in humans. But that's because its function is so critical that we have to all have the same variety. It's actually part of our immune system, and it's one of the most important parts of our immune system for fighting off infectious disease. Okay. But we are evolving at other gene loci. So for instance, just as an example, humans have gotten taller over the last 300 years. Okay, on average. And so there's been some evolution that's been detected there. Okay, so we can be evolving at gene loci involved in height, just as an example that has been pretty well documented. Questions about that? All right. Questions about population genetics or questions for the first exam? Yeah, take.
So you said it's gonna be more like a short answer then?
There is going to be a mixture of multiple choice, maybe some matching, and then some short answer. Usually most exams are about 40% what I call short answer or short essay. When I say short answer, short essay, they are answerable in a few sentences to 2 paragraphs typically. But yes, it is going to be like if you had me for Bio 1 last semester, those exams were mostly multiple choice. This is going to be much more short essay questions.
Would it be possible for you to do some type of practice?
I can think about that. Yeah, those ones that I posted are a pretty good mix of the type of question you can think of seeing on the exam. Any additional practice problems are probably going to be along the lines of where you're still you got to read and interpret the word problem, but you're still going to do the same mathematical processes. Yeah, Max. So homologous structures is where you have trait similarities due to shared ancestry. With something like homologous structures, so with like homologous, you're going to have an ancestor. They have trait number 1. But then there are going to say be some descendant species. So we're not even like, so we're just saying that these are some descendant species from that. But this species 1, this is going to be like descendant species 1. They're also going to have trait 1, but they might have a modified version of it then. Descendant 2, also going to have trait 1, but again, maybe a modified version of it. Descendant 3, descendant 4. Again, they're going to have trait 1, but they might have modified versions of it. It's going to be similar to what was in the ancestor, but a modified version. And an example that we went through of then homologous structures, for instance, is 4-limb structure in mammals, where we have generally all mammals have the exact same bones and the general layout of those bones in their 4 limbs. That homologous structures, again, trait similarities due to shared ancestry / relatedness. Analogous structures, whoops. Those are going to be due or caused to convergent evolution. In the case of convergent evolution, you might have, say, an ancestor over here. You might have an ancestor in a completely different, unrelated lineage. This ancestor might go through some speciation events still. This one might go through some speciation events. But what happens is you might have, for instance, a trait evolves. You might have that trait evolve. You might have a similar trait evolve here one time, but then you have another trait, that trait also evolve a second different time over here. And then what ends up happening here is this descendant has the trait. If it evolved over here in this lineage, this descendant would have the trait, this descendant would have the trait. But again, these descendants would not have that trait. because it's not coming from their common ancestor. And what ends up happening, this is an example where the same trait evolves multiple times in unrelated lineages. And that's going to be convergent evolution. Questions about that? So convergent evolution, this is where a trait similarity evolves in unrelated lineages, unrelated species. And usually that evolves multiple times. And I gave the example of eye structure in mollusks versus mammalian lineages. Those evolved independently of one another. Another example of convergent evolution, and one of the more just wings, like wings in birds versus wings in bats. The ancestor of bats was just, well, some type of mouse-like rodent, and it did not have wings. Yeah.
Which one was the flying squirrel?
Flying squirrels convergent versus flying squirrel versus sugar gliders. That was an example of convergent evolution or analogous structures. Then. Ancestors of sugar gliders couldn't glide. Ancestor of the flying squirrel couldn't glide. They evolved that ability in their lineages independent of one another on completely different continents with no interaction between them. Other questions? Any other questions before we go on? Oops. Yeah,
We have to know the like alpha, beta, gamma, zeta.
This is an example of gene duplications and what can happen when you have duplicated genes. Examples are always useful to help understand also the concepts that we're talking about in class. Do you have to have it completely memorized? No. But the example is beneficial for understanding the concept.
So what does that mean to cause for small populations?
Small populations are more subject to genetic drift. Any type of random change in a small population is a greater statistical change. So genetic drift is just random change of the population. But if you have like a population of billions, one little change is such a small percentage of a billion. So 1 out of a billion, tiny statistical change, probably not going to influence that population too much. But if you have a small population and there's a random change, say it's a small population of 10, and you get one random change, that's a 10% change. That's a big change. So by being smaller, any random changes are more statistically significant. Okay. We're gonna move on, and this is gonna be though exam 2 content. So we're gonna start talking about the origin of species and basically in this unit, we're gonna be talking about what is a species, what is that definition of what a species is. We're gonna be talking about speciation. Speciation are events that produce new species. And we're going to talk about types of speciation, such as allopatric speciation versus sympatric speciation. We're going to talk about how new species arise. What are some types of what are called prezygotic barriers or postzygotic barriers that will result in speciation events? And then we're going to also then transition a little bit into then how do we name and classify species as well, and talking a little bit about taxonomy. Species. Species is the Latin word, like a lot of biological terms. They have Latin roots, but species is a Latin word that means kind or appearance. And what I'm going to give you as the kind of classical or traditional definition of a species is a population whose members have the potential to interbreed and produce viable, fertile offspring. So this is kind of the old school definition of what a species is. Two organisms are members of the same species if they can interbreed and make offspring that are both viable and fertile offspring. What does viable mean? Yeah. Yeah, viable just means it's living and healthy. Fertile means it can reproduce. The offspring itself can actually has the ability to go on and reproduce them. Nowadays, that definition gets more complicated. This is what I call the classical definition because it's all the way back to say like the 1700s when this was formed. Okay. We'll go through a little bit of the history here in a second. Nowadays, it gets a little bit more complicated sometimes, and biologists might be comparing species and determining what is a species based upon morphological data, physiological data, biochemical data, and then also DNA sequences when they're actually looking at this one. But this definition was formed before they even knew what DNA was, for instance. Then. Okay. Again, that traditional definition of a species is based on the ability to produce offspring, not necessarily on similarity then. So for instance, are those the same bird? I'm not even asking you if it's the same species. Is that the exact same bird? No, but they're pretty similar. Do you think that's the same species of bird? It is not the same species of bird. This is two different species of bird that I have in this particular image. Even though they look almost completely identical, this is an example of similarity between different species. And why are biologists saying they're different species? They can't interbreed. So those two birds right there cannot have offspring together. And it's physically impossible for them to have offspring, making them then separate species, even though they look almost identical to one another. Even though they look identical, there's differences in their genetics that makes them unable to reproduce with one another. Now, are these all the same people? No, and we have a lot of variation there. And despite the fact that they don't look all the same, they could potentially all interbreed and produce offspring. And so the idea here is that because they can still interbreed and produce offspring, then they are members of the same species. Questions about that? Yeah.
What if someone was infertile?
That is, yes, that is an issue sometimes. No, and that's where I said the definition of a species is not always nice and clear cut. And by the way, you're going to get sick of me actually flipping the script and giving you guys examples like that throughout the semester. There's always some exceptions to it. Everything's always more complicated. It's not, everything's usually not just black and white, one way or the other. There's always going to be some gray areas or some exceptions to that. Still member of the same species. Okay. Gene flow is central to this idea of a species concept. Gene flow between populations holds the genetics of that population together. And if you hold the genetics, you're also holding the phenotypes and the traits of that population together. So as long as you're having substantial gene flow, As long as you've got a lot of gene flow, it reduces the possibility of speciation events. We're going to get to that here in a second, though. Okay. So we're going to talk more about what makes a species a species. In this unit, we're also going to talk about speciation. Speciation, this is the process by which one species splits into two or more species. In essence, speciation events make new species. Darwin first proposed this idea in The Origin of Species. He proposed that evolution, natural selection can give rise to new species, and that a whole series of successive speciation events is responsible for then all the different species we see and the diversity of life that we can measure. He also went on, and there's entire chapters just dedicated in Origin of Species on speciation. Also just went on to explain not that speciation explains not only differences that are between species, but also why there are similarities between species as well. And the idea here is that if you had, again, an ancestor, and that ancestor then split through some speciation events and produced two different descendant species, that those descendant species are going to be modified versions of the ancestor. So there's going to be some differences between them, but they're also going to have a lot of homologous structures, a lot of trait similarities between them then as well. And so this model of speciation followed by evolution explain both differences between species, but also then simultaneously explains similarities between species. So evolutionary theory has to explain how populations evolve, how new species originate as part of this, because it's one of the predictions from it. When it comes to new species, the formation of new species is also going to hinge on something called reproductive isolation. Exactly what I mean by reproductive isolation? Again, it is going to basically be a little easier to follow along with just thinking through an example. You might have a population. So you might have one ancestral population. But something happens and that ancestral population splits into two smaller subpopulations. And there's something dividing them apart then. So you have some type of population. Over time, it gets split into two different subpopulations. Some type of barrier is between them. That barrier is going to impede those subpopulations from interacting. So something is keeping them apart, so they cannot breed with one another, and there's no gene flow between them. Okay, there's no individuals moving between the populations. There's no gametes moving between the populations. There's no gene flow. Over time, though, what's going to happen as more and more time passes? Those populations are then going to have some genetic differences that start to accumulate then. Okay, so there's going to be genetic divergence over time as long as those barriers stay in place. Okay, and so typically also remember genetic divergence often starts with mutation, and mutation is random. So you're going to have random mutations in one of the subpopulation. You're also going to have random different mutations in the other subpopulation. Now, there's a number of barriers that can keep those two things apart. Those two subpopulations could be just divided by a geographical barrier. Let's just say a river or a mountain range. So the barriers, when I say there's some type of barrier that impedes the population, it could be a geographical barrier. Could be that they are moving between islands in the ocean. So the ocean could be it. But it can sometimes also be biological barriers. some type of behavior is a biological barrier. A mechanical barrier, it could be a temporal, time-based barrier. And we're going to talk about examples of barriers as we go through this unit and how those barriers then result in reproductive isolation. Okay, again, a little easier to understand an example. So I have a population of my organisms. So I've got my population of Pac-Man here. And let's just say that population gets separated into two different subpopulations. So I got my Pac-Man population at the top, I got my Pac-Man population at the bottom. All right, and so they've been isolated, reproductively isolated. There's some type of barrier, so they cannot have intermixing between them anymore. There's no gene flow between them. But let's say under one, then subpopulation is subject to a predator. So I got my ghost predator here. And so they breed over successive generations, but with different selective pressures. So I've got one population of these Pac-Men that it does not have a predator, whereas this population is then in an area that does have a predator. What you're going to typically get is maybe this original population that's in the same environmental context that before it was before. No predators around. Well, it may not change much over time. It may actually stay relatively constant and have low evolutionary rates of change. But the other population, well, that one, because it's subject to the predator, is going to change slowly over time. Okay, and maybe it's evolving camouflage. Maybe it's evolving something to get around that predation, for instance. But it is going to change differently due to those different selective pressures. Over time, sometimes the genetic differences get so great that even if we remove that barrier and we allow them to intermix once more. So say we remove that reproductive barrier, whatever it is, and now my green Pac-Man and my yellow Pac-Man can actually intermix, can actually occupy the same space. But if the genetic differences are too much, they can no longer successfully interbreed with one another, and then they would be separate species. Questions about that? Okay. So speciation. Again, this is going to be the formation of new species. This is going to be where one species diverges and splits into two or more species. Again, this requires reproductive isolation. Reproductive isolation is required for speciation. And it's going to then, once reproductive isolation has been in place long enough, you get speciation then when gene or DNA sequences become different enough to then restrict sexual reproduction. With speciation, speciation can typically occur in two main ways, either allopatric speciation or sympatric speciation. Okay. By the way, allo equals Latin for other. Sim equals Latin for same. And Patrick equals Latin for country / area. Okay, so those terms translate. Allopatric speciation really kind of translates as other country speciation or other area speciation. Sympatric translates as same area speciation or same country speciation. Last thing for today, and I'll pick back up here. Again, when I say allopatric speciation versus sympatric, allopatric or is other country separation, This takes place when a population is divided geographically into isolated subpopulations, and there's no gene flow between them. Okay, I'm just gonna we'll pick back up with this one. Let us say we have a pond or a lake. That pond or lake starts to dry up. And there's maybe a area as it shrinks up, there's an area of land that now separates that pond or that lake into different lakes. Okay, before in that lake, all these fish were together. And they were not reproductively isolated, there was gene flow happening. Here now, once this area dries up, you've got one population and another population, and this area of land prevents them from intermixing. They're reproductively isolated and they're geographically isolated. This is going to be an example of allopatric, but we'll go through some other examples of what this looks like, and then we'll talk about sympatric speciation. You might want to study a little bit ahead on sympatric speciation before Wednesday's lecture, because that's the heart. Otherwise, don't forget exam on Monday.